Okay, Mahesh, I think we are live now. Okay,
Yeah.
so I would like to welcome one and all for this edition of the NSF Wednesday Colloquium. It's actually a special one because it's the final one of 2021, the year 2021 that we had. A very difficult year, of course, on a personal note. And also, it is ending on a positive manner in which people have started coming back to work. And also, we have started to meet on a scientific front. So although it was a tough year in the front end, but it ended on a little bit of a nicer, warmer feeling. So today, just like every Wednesday, we have an eminent speaker for this edition of the NSF Wednesday Colloquium. We have a very dear friend and a colleague, Professor Mahesh Hariharan, joining us from IISER Thiruvananthapuram campus, right from his office. Unfortunately, he could not make the trip because of the issues related to travel still pertaining, and TIFR still sort of warming up to opening to visitors. I hope, Mahesh, you can visit us soon next year when everything opens up well. So before I introduce Professor Hariharan, just me take a minute for those who sort of are joining the Wednesday Colloquium for the first time. The Wednesday Colloquium is one of the oldest scientific forum that TIFR has. It actually brings along all the natural sciences faculty together under one lecture theater for 1 hour, okay, on a Wednesday at 4 p.m. This idea was conceived by our founding director, Professor Homi Bhabha, who believed that sciences cannot be segregated into contexts of chemistry, physics, and biology. They actually ultimately will mix together. And that was the idea, the conception of TIFR itself. So it bears a long history, as long a history as TIFR itself, this colloquium series. And many of us, many of my senior colleagues have hosted this forum. This is actually a pretty sort of pleasant job for me to host this. And I thank all my predecessors in this position. So today we have Professor Mahesh Hariharan, who actually comes to us from the Department of Chemical Sciences in IISER Thiruvananthapuram. Mahesh actually is right now a professor at the School of Chemistry. After completing his doctoral research, which he carried out from 2002 to 2007 in CSIR NIST with Dr. Ramaya, Mahesh actually carried out he did some of the very interesting photophysical work with him at that time, which sort of made him an expert in photophysics, and especially with organic molecules. Mahesh, after his training in Dr. Ramaya's group, moved to actually that was a hub for many of the photophysicists who are now faculty members. Just wanted to mention that all across India. So NIST was a very amazing place for doing photochemistry at that point of time. Mahesh actually, along with many of his colleagues, moved out to the States for his postdoc, and he did his postdoc with Fred Lewis in Northwestern University. And it's here where he sort of expanded his work on synthesis and sort of different biological ideas of spectroscopy. And with Fred, of course, who is a well-known pioneer in looking at electron transfer and sort of charge transport in organic materials and biological materials, he sort of delved into that research front. After coming back to India, he joined IISER Thiruvananthapuram as one of the early faculty members in the institute, and he established his research in understanding the interaction of light with biomacromolecules, looked at, established a group that knows how to do crystallography, and especially of organic materials, and of course developed an entire branch of science in photophysics. Some of his very nice work he will talk about today. On aggregates. So Mahesh is, of course, has been awarded for his wonderful work. He's a recipient of the Chemical Research Society of India Bronze Medal, the Japanese Photochemistry Association Lectureship Award, the Asian and Oceanian Photochemist Award, and also Young Scientist Award. And also he is actually part of sort of in special issues that came out from in for young scientists in Chemcom and Chemistry European Journal. And he was also one of the selected young physical chemists by Journal of Physical Chemistry that selected in 2018 a bunch of around 40 to 50 young physical chemists around the world. So Mahesh has established this wonderful group of scientists. One of the students actually recently visited us for a faculty position. So Mahesh, I give you a warm welcome from TIFR. And I would like now you to sort of talk about molecular aggregates and how this is a very nice way to sort of do Exciton isolation. Okay, Mahesh, could you share your slides?
Yeah. Didi, can you?
Yes, yes, I can see the slides.
And is this thing blocking?
No,
at the
fine.
bottom. Yeah. So thank you, Jyotishman, for kind words of introduction, and also thanks to Jyotishman, Professor Kalobaran, Maiti, Sudiptada, and all the faculty colleagues who felt that I should be sharing some of the results during this colloquium hour. Indeed, a great pleasure and honor to discuss some of the results that we have been working in our laboratory for about last 10 to 12 years since 2009. And this has been a wonderful journey so far in a newly started institute then in 2009. And a lot of good students coming from different levels, such as, right after +2, join here for BSMS program, which is a 5-year integrated program that many of you would be aware of. And then the students from PhD program, and also this year onwards, we have started an MS program, which is for 2 years, instead of having the integrated program, which is MS plus PhD. So we have both the programs running parallelly now, which is just MS alone and the MS PhD, plus the BSMS. So all sorts of wonderful students join in order to work on variety of aspects of the project that we are working here. So to cater to the generalized audience, I have tried making it broad enough so that biologists and physicists, apart from chemists, can carry some important information after the discussion. And I would also like to learn more from biologists, chemists, and physicists who would attend to give new thoughts and interesting ideas in this direction. So let me move forward. Largely, I will be talking today on Greek cross aggregate, and over the next 30 to 40 minutes, I will elaborate the relevance and also the challenges that existed in this particular topic. But before I delve into it, there is also something that we are working on, which is not with respect to exciton isolation, but exciton being mobilized across the stack of aromatic hydrocarbons as much as possible. So there are two aspects. One is you take an organic chromophore, or for that matter, any entity which you supply light, and then take the molecule to the excited state and see whether the excited state can dissipate the energy to have the energy dissipated across a larger number of associated entities that are kept nearby. And this is something very essential in terms of carrying energy from one point to another point, and this is largely we call it as exciton migration. This has its own importance. And then there is another aspect which is really important is exciton isolation, which means that you really do not want the exciton to move, but instead the exciton gets just deactivated to the ground state from that particular entity itself where we are making the photoexcitation. Now, there is all sorts of gradient that one can think of in between these 2 points where maximum migration or maximum exciton interaction to no exciton interaction, which is exciton isolation. And then we have exciton mobility with a gradient that is spread across these 2 conditions. And I would be willing to discuss a couple of isolated examples in order to have maximum exciton migration and minimum exciton migration, which is what I refer as exciton isolation, and then describe the context in which this is important, like why these are important. So let me go forward to discuss a little bit about nucleic acids and the photoexcitation of nucleic acids. So what you see here are guanine nucleobases and adenine nucleobases that are indicated in different colors. And when you have these nucleobases that are stacked in a helical fashion and they are sitting on top of each other, these nucleobases have interaction between them through the so-called pi-pi interactions. And one can imagine that upon photoexcitation of one of the nucleobases, the excited nucleobase has got excess energy, which can migrate from one end of the DNA to the other end through the energy migration or the so-called exciton migration that I have referred. But there is also another possibility that if the energy of the photon that is supplied is slightly above the work function of the guanine, you can imagine that there is a loss of electron that can happen from guanine, which is what we call as photoionization. And that can result in creating a charge on guanine, which can be a radical cation after the loss of an electron. And the radical cation can thus hop from one unit to the other unit, thereby moving the charge through the DNA. And there was a claim in 1989, which was published in Science by a group of scientists from Caltech and Columbia together. And they claimed that DNA can act as a conducting wire, which means that if you are able to generate charge on DNA, it can hop through the DNA pretty much like a conductor. So this can be called as a molecular wire. And therefore, in the next few years, it just appeared that any signaling pathway that is happening in biological systems can be correlated to one of the possible pathways for signaling that is happening in biological systems. Although this became quite a popular finding at that time, subsequently, few scientists who are basically spectroscopists or photophysicists started working on it in order to see whether this is really possible that you have charge that is created on one end of the DNA. Can it migrate so much so that can that be declared as a conductor? Now the finding goes like this, that the charge transport across DNA is purely dependent on the number of nucleobases that are present in them. So if you have about up to 4 nucleobases, the charge can move through DNA pretty much like a conductor. And if you have about 5 to 10 nucleobases, the charge can move pretty much like a semiconductor. And if the length of the DNA is pretty much above 10 nucleobases, let us say beyond 10 nucleobases, then it almost acts like an insulator. So the claim that was made.
Mahesh,
Yeah.
Mahesh,
Yeah.
sorry, Mahesh.
Yeah, please.
There is a question in the chat box.
Sure. Yeah.
It says that is it easier for the electron to move or hole to move through?
Okay, that's quite an interesting question, and sort of not so much answered for the reason that radical anions, when created in aqueous media, it tends to get quenched, so that you really do not see easily the signals corresponding to the radical anions when the measurements are done in water. But at the same time, the radical cations for the nucleobases are also not seen that easily in the context that the nucleobases, radical cations and anions have very low molecular extinction coefficient. So in terms of the transient absorption measurements, if we do, these are species that are not easily seen. Therefore, what has been done is to connect a chromophore that can pull or push the electron towards itself, and thereby tracking the charges on the chromophore is an indirect way of tracking the charges on the DNA. So we really do not see the charges on DNA directly by itself. I think there is a comment that is added by Ravi, who is really an expert in this particular topic. So he is adding that the positive charges are not ionic, but rather holes. Yes. And there is a driving force for the holes to migrate to the red chemical moiety. Yes, exactly. So Ravi answered it much more precisely. That if I have given a wrong impression that it is a positive charge, it is not cation, but instead it is a radical cation that are generated, which moves across the stacks. I hope I have answered the question. If not, I'll be happy to discuss this in greater details. Yeah. So what we see here, with all these going around, DNA is somewhat resistant to the photoexcitation process, which means that DNA do not undergo photo transformations that easily, even with the capability of charges moving little bit around, plus the photo-oxidation possibility that exists in DNA. DNA is self-protected from the charges that can be created. Now, there are two possibilities that DNA can get mutated. One is the G oxidation, which I have referred here. And another possibility is that when two thymines come close to each other, it can form the simple 2+2 adduct that we have studied in cycloaddition reactions, and thereby forming a thymine dimer, which is responsible for melanoma cancer, which essentially comes from lack of melanin in our skin, and thereby allowing the UV light to pass through the skin to reach DNA, to excite the DNA chromophores with UV light, and that can result in two major reactions, and one reaction results in the basic reason for the melanoma cancer, and the other one is responsible for conversion of G to a radical cation, and reacting with water at this position, hydroxyguanine is created, which eventually transforms to 8-oxoguanine, which is also responsible for various other types of cancer that can occur in our body. Now, having said this, there is a proofreading mechanism to make sure that these errors are being corrected time to time by photolyase enzymes or the adjacent environment that DNA has in order to protect such kind of transformations from the incidence of UV light, thereby creating very low quantum yield of these reactions that can happen in our body. So this is the DNA's way of protecting itself, and perhaps this protection of DNA not being so reactive and not much charge productive comes from the fact that the DNA has a helical twist between the chromophores, which results in reduction of electronic coupling between the nucleobases, and therefore do not sustain the lifetime of the charges that can be created in it. So this is one aspect that I wish to discuss, is the selection of nucleobases as well as the selection of chromophores is quite important as to what is required upon photoexcitation. The second aspect is the arrangement of the chromophores. If two of the chromophores are arranged, facilitating them to have more charges that are sustained in the stack, thereby you can have more reactivity that is generated from the biological scaffold. So the first example that I am discussing is the DNA by itself, where the attempt is to minimize the redox states that are generated, and yield wise as well as the lifetime with which these charges are being generated. The second example, in contrast to the nucleic acids, is the photosynthetic reaction center, wherein one can see that the chromophores are arranged at the photosynthetic reaction center, is to optimize the formation of the charges, and these formation of charges will thereby help to have the oxidation and reduction happening in the solution of carbon dioxide and water that we have, finally resulting in sugars that are being synthesized. Now let us focus our efforts on understanding the photosynthetic reaction center, wherein we have two chromophores that are sitting exactly as a cofacial stack at the center of the photosynthetic reaction center. And you can see there is a subtle difference between nucleic acids and the photosynthetic reaction center in terms of the arrangement of chromophores. In the other case, it was a helical stack, and it was more of a symmetrical structure. Here also symmetry exists, but not as symmetric as DNA. And you can see here that the protein scaffold holds the chromophores in a manner that initially two of the chromophores are well interacting, and that goes to another chromophoric unit which is not as much interacting as first two, and then it goes to a non-metallated porphyrin, and then a quinone, and then another quinone that you see in this entire scheme of things. While in the case of DNA, there are only 4 chromophores that you can see, which we know as G, C, A, and T. While in the case of photosynthetic reaction center, there are mix of chromophores with metallated versus non-metallated porphyrins, and also having quinone in it. And these are all different layers of energies with respect to the chromophores that they possess. And by creating the photoexcited states, the electrons move from one unit to the other until it receives to the quinone that is indicated over here in blue color. So let us begin with the first step, which is called symmetry breaking charge separation. And towards the end of my lecture, I will be elaborating a bit on what is the importance of symmetry breaking charge separation in the context of what we are doing. But to begin with, the photosynthetic reaction center has nicely optimized to have 2 chromophores that are cofacially arranged. So in an ideal situation, 2 of the chromophores, which are same in its constitution, upon photoexcitation should not be sharing a charge from one unit to the other unit. But then one can optimize several parameters that are related to 2 chromophores to have an instantaneous difference in solvation, and also various other parameters that one can think of. So at least one case is the instantaneous solvation that exists between these 2 units, so that upon photoexcitation, an electron will jump from one unit to the other unit, so that one creates a whole electron pair that is generated between these 2 units. Now, the excess electron that is generated will move from this special pair to the next unit, and further to this unit, and to this unit, and then finally to this unit. And nature clearly understands the application of Coulomb's law, So if you have two charges that are generated over a very short distance, you have 1 over r dependence on the charges that are created in terms of its energy of interaction. So you can imagine that at a shorter distance, they can easily recombine. So nature plays a trick in order to have the sequential electron transfer jumping from one unit to the other unit until it reaches to a distance which is much larger than the distance to which initially it got generated. So you can see that this distance is much more. So therefore, the Coulomb's interaction gets reduced, and as a result, those charges will survive for longer time. And so long as we get the charges for longer time, those charges will interact with the environment in order to use them for catalytic activities. And that is the purpose of this optimization of photosynthetic reaction center. So these are the steps that are characterized it to be sequential electron transfer going from the whole electron that is getting generated here. And finally, this electron would end up in this particular quinone, thereby having a larger distance between the hole and electron, and thereby longer survival time for the charges. Now you can see in both these cases, let it be DNA or the photosynthetic reaction center, the selection of the chromophores and the way that is arranged makes a whole lot of difference as to create the excitons and making it charge separated states, whether these charge separated states should live longer, whether the charge separated state should have sufficient quantum yield or not. Depending upon what we are interested in that context, we can pick the chromophores the way that we want and arrange them in order to get the function that we are interested in. So these are two interesting biomolecules that we would like to take motivation from. And this is a slide that narrates different pairs that exist in purple photosynthetic bacteria, photosystem 1 and photosystem 2. And you can see these are the special pairs from the center that has been picked and the distances that are indicated over here. And the electronic coupling that is evaluated for these three pairs are so much different that at the end, all these photosystems are very efficient in catalyzing the photosynthesis. So it clearly means that,
may I ask a question?
Hello, yeah, there is a question, sorry.
Yes. So,
yeah.
So this photosystem, the description of the distances and the rates. So how dynamic are they, or are they like very static, like as in crystal structure kind of?
This is quite an interesting aspect. They are buried in protein scaffold, which means that largely they are static. But then upon photoexcitation, certainly the reorganization energy depends on the components that are involved in the photoexcitation processes. For instance, the chromophores that are naturally selected by photosynthetic reaction center have minimal reorganization energy and maximum electronic coupling, so that there is a charge transfer component which is significant in all the three dimers that are shown over here in the overall scheme of overall electronic coupling that exists between them.
No, but even in the ground state, they are like very static structures, or is there like a ensemble of different structures which form these pairs of porphyrin like systems?
Okay, so in different photosystems, for example, in photosystem 1, this is the only dimeric arrangement that is available. There is, I think your question is in the direction of whether there is a distribution of structure between the special pairs that exist.
Yes.
But it is only one. Like, the special pair has a fixed geometry in photosystem 1. In photosystem 2, it's slightly different, but still having orbital overlap between the chromophores. And in purple photosynthetic reaction center, this is the arrangement that exists in it.
Okay. Thank you.
Sure. So moving forward, to have chromophores arranged in a designated manner, and to have an understanding on what is the overall electronic coupling between the chromophores is a topic of interest by itself at the moment in two directions. One is to understand as such in the context of the natural photosynthetic reaction center, and in another occasion, it is in the context of in the context of the artificial systems that one can create. Yeah. So moving forward, we in our group work on three major aspects to understand the photoexcitation processes in aromatic hydrocarbons. Just to see, like, what happens upon photoexcitation and thereafter. And we also try to bring in chromophores from different types of chromophores into interesting architectures that you can see. So having discussed the photosynthetic reaction center at length, this particular structure which I am showing here could be reminiscent of the photosynthetic reaction center, but this is in the crystalline lattice that we have generated. So this is a radial distribution of chromophores. And then this precisely characterized structures which are organized the way that we are interested in. Once we achieve that one, we try to photoexcite them and understand the photoexcitation processes in the context of the aggregates, which Jyotishman was mentioning in the beginning of the interaction. So moving forward, these are three detailed reviews and accounts that we have written based on some of the contributions from our group and then the contributions from other groups as well. And it can be classified into population of triplet excited states, which we are quite interested in because it's a long-lived excited state. And then it's a neutral state. And then from there, can we generate long-lived charge separated state? And to achieve them, can we have unique chromophoric architectures, imbibing motivation from the natural photosynthetic reaction center? We try to create different type of aggregate structures to study them. So I'll be explaining one example each, if time permits, from each of these three topics, although the major emphasis will be on the last one, which is unique chromophoric architecture, which is what we call as Greek cross aggregate. Yeah. So one quick question that we would like to discuss today in the context of the charge separated state is, can we make non-parallel stacks of donor and acceptor units? And if yes, do those structures show long-lived charge separated states? Now, these are quite simpler things in terms of crystal engineering at the moment. Taking an aromatic hydrocarbon, just do any sort of functionalization in the periphery. Each functionalization would end up transforming the crystal packing of the aromatic hydrocarbon. And this has been established for quite some time. And in 1989, again, interestingly, it's the same year as I mentioned earlier for Jacqueline Burton's contribution. In the same year, there was a very nice review by Deshi Raju and co-workers from India, reporting the classification of aromatic hydrocarbons into 4 major domains. One is herringbone, another one is sandwich herringbone, and then the third one is brickwork-like arrangement, and the fourth one is a columnar arrangement. So you take any hydrocarbons, you can classify them into one of these 4 architectures. And over a period of 30 years, this is well documented as well. But then if you have one more component connected to it, which is a donor and an acceptor attached to each other, you would see it's a whole lot of complexities that arise. One is like, you take and simply mix donor and acceptors, which are electron donors and electron acceptors. They tend to form cofacial donor acceptor complexes, which we see in our undergraduate program. If you wish to figure out whether you have aromatic hydrocarbon in solution, you add a little bit of picric acid onto it, you will get nice crystals formed. And thereby we say that the aromatic hydrocarbon is present in our sample. And that's essentially because of the co-crystal being easily formed between anthracene, and picric acid. Likewise, it forms with various other aromatic hydrocarbons. And typically, those kind of cofacial donor acceptor structures are called Iverson's poldamers, as you see over here. And if you wish to make any alternate structures of organic structures, meaning that, if you have stacks of acceptors and stacks of donors like this, it's an extremely challenging task. Takuso Aida and co-workers have achieved a great deal of control over making such kind of heterojunctions. And in India, Subi George and Arindam Chowdhury have contributed a lot in this direction, characterizing such structures and understanding the detailed properties of those assemblies. Now, there are several other attempts being made, making monohelical stacks of donor and acceptor, double helical stacks of donor and acceptors. This is reminiscent of DNA structure, as you see here, but with different chromophores, not the nucleobases G and C and A and D, but instead it can be naphthalene and perlene, etc. Now, there were attempts to make parallel columnar structures, as you see here, the stacks of donors and acceptors. And then we were wondering whether we can make twisted stacks of donors and acceptors. And our interest was initially little bit disappointing to understand that if you have two chromophores that are connected to each other, they would not crystallize. Instead, they would form amorphous architecture. And this is the diagrammatic representation that was published in one of the papers in 2012. Surely, like the students who were working at that time took this for granted and would come back by telling that, no, this compound is not crystallizing. Until one of the students in our group came up with the feeling that, these are tiny little crystals that are forming, but not diffracting at that moment. Yeah. So, but then in 2012, we saw that it's the same compound that was shown in the previous slide got crystallized with different orbital overlap that you can see between the PDI chromophores. And these are in turn crystallizable, even if they are connected to each other in this manner. So our group resorted to getting such kind of structures between the donor and the acceptor. But then there is a problem of heterogeneity that is possible, even if we are able to stack them on top of each other. And the heterogeneity comes like this, that all the acceptors can sit on top of each other and all the donors can sit on top of each other.
Mahesh, a quick question.
Yeah.
If you go back to the previous slide, could you tell us these twisted sort of PDI structures that you had, dimers, they can form very easily crystalline films, or they have been crystallized here? And whose work is this? Whose work is this?
From a Chinese group, and they have crystallized it, and there are it can be easily converted into thin films.
Okay. And you showed H and J type both. So they just require a little bit different crystallization condition?
No. This is actually within the given crystal structure.
Both is seen.
There are two forms that exist.
Okay, fine. Thank you.
Now, taking this further, you can see there are two possibilities of having the donors and acceptor stacks, quite self-explanatory in this slide. We started working on this topic. Can we predict which out of the two structures will be formed when you have a donor and acceptor? It is not fully resolved to understand which out of the two will form. So I'm just throwing it as a discussion topic here as well. So we took from CCDC a set of crystal structures that are formed between TTF and TCNQ. This is a pair that is known to have a very strong tendency to form co-crystals. And we took about 19 co-crystals that were available with different substituents that are present in the periphery, and could figure out that depending upon the substituents, some of them tend to form donor-on-donor, acceptor-on-acceptor stacks, while some of them tend to form the mixed stacks that you are seeing here. Given the same chromophoric combination, which is the electronic donor and the acceptor, which is TTF and TCNQ, just by changing the substituents, it changes the packing, and you can see that the property of the material goes from metal-like to semi-conducting to almost insulating in nature. Yeah, and this in different forms of all these structures have been established in the literature. We haven't contributed anything to it, but we wanted to figure out using symmetry adapted perturbation theory whether the energy fragmentation that we get in terms of Coulombic polarization, dispersion, and repulsion that you can isolate in terms of the interaction energies between the units. Can we make any sort of a correlation that by putting a particular substituent, can we end up getting this particular structure over the other ones? It is disappointing to say that we could not get any sort of correlation, but instead we figured out that it is still more of an empirical way of getting such structures, and it forms the donor-on-donor, acceptor-on-acceptor for the structure that we get, but then in that case we get repeated. Yeah, so we went ahead in order to work on connected system. This is purely synthetic effort, and then we made donor-acceptor system, about 116 of them, a big library, out of which 17 of them got crystallized, and 2 of them ended up getting structures that we see here, which are donors stacked on top of each other and acceptors stacked on top of each other. Now, what is so important about this stacked structure is that, let us assume that we are photoexciting this donor over here. The excess energy allows an electron to jump from the donor to the acceptor, now thereby creating a charge separated state, but charges are now within the vicinity. Now the charges can immediately recombine, all within 110 femtoseconds, which is the pulse width of the measurements that we do. So anything faster than that is something that we cannot capture. So in this time scale, you see that the charges separate and recombine. So it is pretty much instantaneous in nature in our measurements. While if you have the stacked donor acceptor system, the charge separated state thus created can delocalize in order to pass through the stacks and have a long-lived charge separated state created. And this was our original thought. And this has also been seen by various other groups in the past, but not been studied on a twisted stack of donor acceptor to see whether the long-lived charge separated stacks can be created. So in our measurements,
Mahesh, can I ask a question?
Yes, please.
Mahesh, yeah, this is always a very interesting architecture ever since you first showed this actually. But one thought has always remained with me actually in terms of the delocalization lengths of the donor and the acceptor states.
Yeah.
Right. So if you look at the electronic states, how localized are these states? I mean, when you excite, are you exciting one of these donor chromophores, or is it like a state which is delocalized across the whole donor system, for example?
This is quite interesting. I mean, from the ground state characteristics, we presume that the delocalization is not much, meaning that, we tend to see that the characteristic of the chromophore, even in the aggregate, is pretty much retained. But then this is a tricky thing, because in the context of aggregate, whatever exciton interaction will lead to, in some chromophores, it results in very little changes only when they are in the aggregated state. So this is something that we have not clearly quantified to answer your question. So my presumption would be that, we are exciting a stack of chromophores, and then having the delocalized charge that is spread across the stack. That's what my guess would be. But this is something that we can discuss more in order to figure out that, how precisely we can figure it out. We are now making a naphthalene-based system in our group to have only 2, 3, 4, 5, and 6 naphthalenes in the vicinity. Probably that will shine little more light into the question that you are asking. But the problem there is that they are not stacks. They are oriented in random directions.
Right. I see.
Yeah.
Okay.
So it's challenging to get the system made in order to do such experiments. Dimer is easy, but trimer is a challenging thing. So sorry.
Mahesh, added to Ravi's question, I mean, and what you were just saying, has anybody tried, like, because there are these morphs and, coughs, has anybody tried to then, put donor acceptors in a very regular arrangement in coughs in a particular distance and see how that would work?
In fixed distances, I have seen, I mean, from TAFR itself, JD has contributed a very nice work. But changing the distances is something that I have not seen with respect to the chromophores by tuning the spacer length or so. Often what happens is that when you change the spacer length, to get the crystal structure in each of those cases are quite challenging. But indirectly from spectroscopic measurements, one can say that we are able to tweak the electronic coupling and therefore the resultant photophysical properties.
Okay.
But precisely speaking, in supramolecular assemblies, the spacer dependence is something that I have not seen yet. But that is something that is interesting as well.
Thanks.
So, so this is the result of what I have discussed. So when we have a single unit of donor and acceptor, there can be charge separated state and the recombination. And in acetonitrile solvent, that's what happens. So we don't see any sort of signal corresponding to the redox states in the excited state. So in chloroform, when they are aggregated, all the naphthalene sitting on top of each other and all the naphthalene sitting on top of each other end up having very nice signals corresponding to radical cation of naphthalene, radical anion of naphthalenimide, and also radical cation that corresponds to the delocalized radical cation in naphthalene dimer. And this is a signal that was long ago established spectroelectrochemically by Kochi and co-workers from University of Madison, where they precisely made a cyclophane with naphthalene sitting on top of each other and plug out an electron from it. And the signal that corresponds to the radical cation of naphthalene dimer pretty much overlays with the signal that we got. And this has a very long-lived state, which is of the order of longer than 1.2 nanoseconds, as opposed to an instantaneous charge recombination that one would see in this particular case, which is the donor acceptor system when in isolation. Now, this brings to a topic where maximum exciton isolation is something that we were, maximum exciton interaction is what we wanted to discuss today. And now I will move on to a topic which is related to exciton isolation, taking the remaining part of my talk. So let me do a quick moving to a slide which is on the topic that, I'll be discussing for the rest of the time. So the first one was to create as much charges propagated through the stack, and the second one that I'm going to discuss is to retain the photoexcitation in a particular chromophore. So for this, what we have done initially is to see the chromophores, or rather crystal structures that are reported in CCDC. So you can see here over the years, 1.1 million structures are deposited in CSD, and that comes with different types. One is, one classification is organic versus metal organic. So one can say that it's purely organic versus inorganic complexes, or organometallics or coordination polymers, etc. All of them classified under this. Another way to look at it is not polymeric versus polymeric. And the third way is to figure out single component, which has only one particular species, and multi-component, and it can include solvents or co-crystals, etc. In all these, what we could figure out is that whenever aromatic hydrocarbons crystallize, they crystallize in one of the forms that I have narrated in the beginning, which is either herringbone or sandwich herringbone, or brickwork, or the columnar stacks, which was classified by Desiraju and Gavaswamy. While we wanted to figure out whether chromophores can exist in cross stacks. So one of the first thought that will appear to our mind is that when you take few books and try to cross stack them, it is certainly not space efficient. So anything in nature, it's space optimized. So it would like to restructure itself in order to make stack of books, which will be much more compact and not cross. So thinking in that line, we wanted to know whether such kind of chromophoric arrangements are at all possible. So we did a lot of search in CCDC to figure out that there are none, or our search is not optimized to find them. Either way, we could not get that. We are still optimizing our search in order to figure out that. Now, that was the time when we were also working on strange architectures in our group. So we managed to get a hexahalosynthon, which means that 6 of the halogens that come close to each other, in order to create a structure that is 6-fold symmetric that you see over here, with naphthalene chromophores that are radially assembled to each other. And this has its own interesting charm of triplet excited state in the assembly. And then this scaffold is an electron accepting scaffold. It can take up an electron from donor and create a long-lived charge separated state. And that is something that we are still working on in order to mimic the photosynthetic reaction center in our. But then today's discussion is mainly on a topic which is null exciton, or rather aggregates by itself. So let me get started on that. That aggregates are being studied since 1909. You can see the first literature that is reported here for isocyanine dyes at very higher concentration. This this group saw that there is something unique and different in the solution at a higher concentration when compared to the dilute concentration. But at that time, they could not figure out that there is something called aggregate, or the aggregates are doing such kind of optical properties that are exhibited. And about 30 years down the line, Scheibe and Jelly independently started working on these type of concentrated solution and came up with an argument that it is an aggregate that is creating the difference in optical properties that are seen. And this J that you see here is from where the J aggregate is termed as. However, these are two equal contributors to the topic. In some places, such kind of aggregates are called Scheibe aggregates, and in some places it's called Jelly aggregates. However, for convenience here onwards, I would call it as J aggregate, which is from this particular literature. Now, it was again 30 years from then, Exciton model in molecular spectroscopy got evolved by a very famous scientist, Kasha, and we study Kasha's rule in order to understand the dipole-dipole interaction. And this particular theory, or rather approximation, gives the the interaction, a picture about the point dipole approximation. So you just take the molecule, it has a transition dipole, and the transition dipole interacting with the adjacent transition dipole. One can pretty much explain why the spectral shifts are seen in any aggregates when compared to the monomeric chromophore. And this was a proposal that was made by Kasha and co-workers in 1965. And since then, this has become a very important paper for those who study the interaction of chromophores, let it be in assembled state or crystalline state or in protein scaffolds, or no matter what. This particular theory is a very well accepted a model for understanding chromophore interaction. So let us see that the dipole interactions have been proposed for various types of aggregates that can exist. It starts from J aggregate, where the transition dipoles exist with a translational offset. Moving on to H aggregate, where the translational offset is minimal. They sit exactly on top of each other. And what has not been studied very much is a rotational offset between the transition dipoles. And that's what you see over here. Although this has not been experimentally studied much, all the theory has been worked out by Kasha. And the idea of H and J aggregates have been extended to the experimental groups in order to interpret the spectral shifts using the model proposed by Kasha. And still, this is a very valid model, though approximated. The better models are slowly evolving. And I would like to narrate some detailed description of what was missing in Kasha's point dipole approximation, and what was, we could not say that it was missing. Kasha in his original paper also proposed that there is an important component called charge transfer interaction, but he did not consider evaluating them, but instead tried proposing the aggregate characteristic based on point dipole approximation. However, over the years, when the tools became easier to handle, and several theory groups came up with the proposal of charge transfer interaction also being included into the model, with an overall descriptor of the coupling with a linear combination of the Coulombic coupling, which was proposed by Kasha, and the charge transfer coupling, which was also proposed by Kasha, but then evaluated in a manner that can be extended to the aggregates that we study. So here you see the dipolar coupling can be evaluated for various displacements, translational versus rotational offsets. And then when two transition dipoles couple with each other, this is the symbol form for the Coulombic coupling that one can write down, and then evaluate the J pool, which is the Coulombic coupling for two transition dipoles interacting with each other. But why this model has certain shortcoming is narrated in this particular paper quite effectively. Though this was a chancy crystallization, that two of the chromophores ended up at a translational offset of 54.7 degree, wherein two of the transition dipoles are expected not to impact. And this is what we typically call as magic angle. It's the same magic angle that we study in Karplus equation as well. So you can see that when these two transition dipoles are at 54.7 degree, and if the two chromophores are interacting only through dipolar coupling, then in this crystalline state, the optical signal should have been matching clearly to that of the monomeric state. But in reality, when they measured in these crystalline state, the monomer and the crystalline state has huge spectral shifts, which means that apart from the dipolar coupling, there is something else also that is operating in order to contribute to the spectral shifts that are seen from the dimer to the monomer. And this resulted in deeper understanding of the entire process. Apart from the dipolar coupling or the Coulombic coupling, several groups have started studying charge transfer coupling as well. So how does the charge transfer coupling occur? You can see here, from two of the homos overlap, the whole transfer coupling arises. And if two of the LUMOS overlap, the electron transfer coupling arises. And this is so, the product of this could lead to a meaningful parameter, which is defined as overall charge transfer coupling. And this charge transfer coupling in linear combination with the Coulombic coupling can give a description about the overall electronic coupling. So if this is a little too much, I can go in steps as we move forward in order to explain them in greater detail. However, you could see that the charge transfer coupling, which is resultant of the whole transfer coupling and the electron transfer coupling, were thought to be in the last decade. The groups like Bredas and co-workers reported that it is sensitive to the relative positions of the molecules. Bernd Engels and co-workers proposed that it is very sensitive to even small molecular displacements. And Alessandro Troisi reported that it is extremely sensitive. And also Clemens Kormenboeuf reported that it has complex dependence of charge the transport characteristics. Now, putting it all together, the conventional thought in early 2010 to 12 is that it is sensitive and unpredictable. And that is when Franz Pano and co-workers came up with a very nice scheme of evaluating charge transfer coupling as well as the Coulombic coupling, and then add them together to get the overall electronic coupling between the chromophores. And this has changed the understanding of aggregates in a completely different manner, meaning that anytime two of the chromophores that come close to each other, the interpretations were based on the point dipole approximation or the Coulombic coupling that was proposed by Kasha and co-workers about 50 years ago. But since this report has come, the idea is to comprehensively understand the aggregate characteristics in the context of the overall coupling. So there are several groups who have started studying keeping the chromophores at various angles. And this is the motivation that we had in order to see whether chromophores can be kept cross to each other. And when I define this as recross, it means that any offset of one of the transition dipoles over the other axis would result in Latin cross, which means that the top portion will be smaller and the bottom will be larger. Instead, what we are talking about is a symmetric cross structure, and that is what is the Greek cross structure is. Now, this when computed for its interaction energy by the groups that are indicated over here, could find out that more favorable ones are the parallel stacks, and the cross stacks may not be that stable. This is in the context of liquid crystals, and this has been studied long ago. So that also means that if you take the overall population of the crystal structures that are reported in CCDC for aromatic hydrocarbons, the same rarity for the cross structure should exist, just because of the fact that the interaction energy is much less for the crossed architecture as opposed to the stronger interaction energy that may exist for the parallel stacks. That could be the case. And then we started digging up the literature, which are the groups that have studied any sort of rotational offset that is possible in the chromophoric aggregates. So this is the literature that I have already shown you, that there are two chromophoric systems that are at an angle of 54.7 degree, and that results in no Coulombic interaction between them, but significant charge transfer interaction between the two units. Yeah. So thereby implying that charge transfer interactions have to be well understood. So these are the efforts that have been made from various groups with respect to rotational offsets, 10 degrees between the transition dipoles, 40, 62, 60, and it keeps on going. So I will flash those examples just to give you a feeling which are the chromophores that are studied and what are the angles between them. Now, this is one attempt by Mullen and co-workers from Max Planck Institute in Germany. They have studied liquid crystals of a very large aromatic hydrocarbon crossed to each other, characterized using solid state NMR in the liquid crystalline phase, but have not explored any sort of photophysical properties in this context. Now, this gives little bit of a thought that there is a possibility to have chromophores cross to each other. However, in the crystalline lattices, if one explores, you would notice that we get the crystal structures which is having the transition dipoles having 99 degree between the two units, 85.6 degree in Berlin diamide with bulky substituents on either side, is also forming cross Darkist or architecture as you see here. Now, there is also a possibility that two chromophores can sit on top of each other with 70 degree, reported by Yuguang Ma and co-workers. And then I would move on just flashing these examples. But at the end, in order to come to a molecule which has two Berlin diamide reported from Frank Wuttner and co-workers, this is quite an interesting study that one would not often think this as a possibility. So this compound, when put in solution, has all the properties, all the photophysical properties similar to that of the monomer of this unit that you have here, which means that you tend to doubt whether you have prepared a dimer in this situation. But all the spectroscopic characterization show that it is a dimeric unit. But then in the electronic properties measurement, one sees that it is very similar to the monomer characteristic that you have in this particular system. And this is well explained by Frank Wuttner and co-workers, proposing the thing that this has a very strong Coulombic coupling, which comes from the orbital interactions between the two Berlin diamide units, and then comes from the dipolar interaction between two Berlin diamides, and then a very strong charge transfer component that you see here, which is roughly equal in magnitude, 545 and 555, but then opposite in sign, thereby cancelling them out in order to get a value which is close to zero in terms of the overall coupling, and thereby having the dimeric properties, the photophysical properties, which are comparison to the monomeric photophysical properties of the chromophore. And this was the first case where experimentally a null exciton was reported, where the pure descriptor of the Coulombic coupling is not sufficient in order to explain the spectral shifts that are there in the aggregates, but instead the charge transfer coupling also have to be included. So you can see here the total coupling is nothing but just a combination of Coulombic coupling and the charge transfer coupling. And the charge transfer coupling is computed using this particular expression that you see here, in which hole transfer coupling and electron transfer coupling is something that we have already seen in the previous slides. It was then our group was working on the aspect of having chromophores that are crossed to each other to see whether precise cross structures can be achieved in the crystalline state. And we were lucky to have this dibromo Berlin tetraester that is seen here, has a crystal structure which has the chromophores that are precisely on top of each other with a 90-degree angle between the transition dipoles existing between them. We went on to see how the spectral properties look like in solution versus the crystalline state. And we can see here with a little bit of perturbation, the signals are almost overlaid with the monomer and the crystalline state for the electronic absorption, emission, as well as the fluorescence lifetime. And this clearly tells that the chromophores that are arranged and cross on top of each other, upon photoexcitation, the excitation is retained in one chromophore and it is not now dissipating to the adjacent chromophore. And that is what we meant by exciton isolation. And by keeping two of the chromophores cross to each other, it is quite understandable that the exciton is isolated and it is not now migrating. And this is definitely an example that is opposite to what we have discussed in the beginning, where maximum exciton delocalization is what we have expected there for creating long-lived charge-separated state. While in this particular case, whatever is excited onto the chromophore, it gets retained in that particular chromophore. Thereby,
Mahesh, Mahesh, quick question.
Sure.
The solution data that you say, the solution data is monomer molecules or these dimers?
Monomer.
So all are monomers, right?
Yes, yes.
And the crystal is a sort of a dimeric arrangement you have just shown, but it is actually an entire extended system where these dimers are predominant.
Thanks for asking that. I should have explained in greater detail. So this crystal structure has only one distinguishable dimer, and it is an infinitely periodic arrangement of the same dimer that you see for the entire crystal. Right. However, in an earlier case where you asked a dimeric PDI that was crystallized from other group, there you saw two of the overlap where you have distinguishable dimers in two different fashions.
Sure.
Yeah. So luckily here we had only one. So,
right.
Thereby,
and so when you worked with the crystals, did you cut the crystals to do this? Because they must be very colored, deeply colored, right?
They are transparent crystals, and they are very thin that we can do the
microtoming. You did microtoming or something?
No, it's just microcrystals as they are crystallized from solvents. They are very tiny.
They are microcrystals. Okay.
They are microcrystals. Not
and then you do all spectroscopy like with that crystal mounted on some.
Exactly. Alternatively, what you are proposing is another way of doing it. If one can grow very large crystal,
yes,
then you slice out one of the sections and then do a single beam absorption measurement,
right?
Which is more accurate, but that is not something that we have done here. Our measurements are on collection of microcrystals that are kept between the cover slips.
Okay. Mahesh, actually, I have to tell you, we have 5 minutes actually.
We're done.
Okay, sure.
Yeah, sorry to have prolonged. Yeah. So, so this brings to a point where we have taken an example in solution to have a chromophoric pair that are crossed to each other, sitting like this, as it is indicated in the example C. And the crystal structure is what is indicated over here, where absolutely no interaction between the two chromophores with respect to the dipolar interaction, which is what we call as Coulombic coupling. And then there is a negligible contribution from the orbital interactions as well, which means that it doesn't have any charge transfer coupling as well. Now this, upon photoexcitation, we could see an interesting aspect, which is symmetry breaking charge separation, and the signals that that are corresponding to the redox states are shown over here in polar solvents, but not in non-polar solvents, which means that the polar solvents assist in creating the charge transfer with a meager hole transfer coupling that is present in this system, with almost negligible electron transfer coupling. The overall charge transfer coupling being very small, but still there is a possibility of charge transfer that is existing between these two units, resulting in a charge separated state. But by virtue of an orthogonality between the chromophores, the charge recombination becomes extremely slower. As a result, we ended up having a very long-lived charge-separated state generated from this architecture. So you can see here the chromophores are precisely 90 degrees to each other. The created charge separated state in this chromophoric dimer. This could also be reminiscent of the symmetric pair that we began in the photosynthetic reaction center, where it was cofacial. Now it is orthogonal to each other and creating charge separated state, and through slow recombination, it gets to the ground state. And this also says that, why nature has chosen a particular arrangement of dimer as opposed to other dimers. And these are somewhat newer models that are coming up in the literature to see why nature picked the cofacial arrangement in order to get the symmetry breaking charge separation. So we are also working on some other angles between the chromophoric dimers in order to get more insights on it. So with that, I'm on my final slide, thanking all those who have contributed to the work that I have discussed today, starting from Rijo, who was my first PhD student, to all the students who are shown onto the left, and a number of BSMS students from IISER Thiruvananthapuram who have worked in the group in order to contribute to the work that is presented. Support from various funding agencies and also former and current directors of IISER Thiruvananthapuram. With that, thanks for your attention, and I'll be happy to answer any questions.
Thank you, Mahesh. Thank you very much. I request all in the Zoom as well as in the audience to give a round of applause. Thank you, Mahesh. So this actually was very interesting journey that you started from the ideas of photosynthesis and charge separation to coming to aggregates where nothing is moving. Everything is isolated. In some sense, the exciton is localized. So even in an aggregate geometry. So thank you for that very nice tour. I'm sure there are questions. So I would request the audience, both in the Zoom and if there are any in the lecture theater, to ask. I have questions, but I'll just request my colleagues to unmute themselves and ask questions. Questions for Mahesh? Okay. If there are, okay, Sudipta, yeah.
There are some in the chat. You take them first, I think.
So
the chat, I think we
Ravi
did some. Okay, Neha, very nice to talk about a very naive question. What are the applications of such systems? Neha, a postdoc from from BCS is asking. Yeah.
Neha, thanks for the question. So when you say such system, I presume that you are asking the two examples that I have
Aggregates.
discussed.
Yeah.
So the first one, the aggregate having long-lived charge separated state is something that is being sought after for the photovoltaic devices, while the second one, the crossed architecture, is a sought after system for solid state lighting purposes, meaning that many of the chromophores are bright fluorescent when they are in solution and existing as monomer, but then when they aggregate, they lose the fluorescence. And there is altogether another paradigm where people work on aggregate induced enhanced fluorescence, but then this cross architecture can be aggregate induced but then retention of the fluorescence. So you have designed the monomeric properties of the fluorescence, but then if you are able to cross it, it has got the same properties in the aggregated structure, but the properties are from the monomeric units. So they are quite good for the lighting applications in the solid state.
Thank you, Mahesh. Question from Sudipta.
Yeah, Mahesh, another beautiful talk I heard from you. Thank you so much. I have a general questions. So you are trying to optimize the geometries of these complexes to get specific properties.
Yeah.
So you start beautifully from the structure of the special pair and go into the things that you have created.
Yeah.
Now, one big question for the photosynthesis side is that the special pair sits in a scaffold.
Yeah.
Okay, and the scaffold both, of course, maintains its structure, but also gives it some dynamics. And the age-old question has been how much in each system the dynamics couples to whatever nature is trying to achieve.
Yeah.
When you go back to structures like this, what you're right now on your screen, on our screen, that part is missing. So do you think that's, would you like to comment on that? You know, this is essentially different from what nature is.
Yeah, very nice question, Sudipta. I think there are two contexts that I have discussed. Possibly, I have mixed it up. So, and probably the last one I went a little faster. So this is where we try to study them. So one part that I have discussed is in the context of the crystals. So for example, this one, while this particular case that we have studied is dropping it in solvent, which means that it allows all sorts of dynamics that it can go through. But the dynamics is not so much so that any alkyl chain between two chromophoric units will offer, but the dynamics is retained with certain angular displacement between the two chromophoric units. So there is some room for the molecule to reorganize. It's not as rigid as we think in the crystalline state, but then it is not like fully movable. So this example that I have discussed is in solvent, in toluene, in THF, and in acetonitrile, and they have some sort of dynamics associated. And slowly we have started making pre-oriented chromophores, dimers and trimers, with which can be put into solvent and still retaining somewhat the structure that we are attributing them to, but then with allowable dynamics that is associated.
Yeah, if I can just clarify my question a little bit more. So yeah, in solvent, of course, you allow all kinds of dynamics.
Yeah.
In crystal, that's much more limited.
Yes.
But in a protein, it's kind of design dynamics possible. There are protein parts which can move as a response to the excitation or to the charge transfer, which can help separate and stabilize the charge transfer states and all that. So this sort of is the core of it, which doesn't have that scaffold, kind of a smart scaffold. And in some cases it's smart, some cases actually it doesn't matter. People are so I can imagine that starting from here, you might think of building a minimal scaffold, which will help some of them. Neither too fluid nor absolutely rigid, but absolutely right modes, which are allowed in that.
Okay. I get your point completely. And, so now this brings to an idea that is well debated, but not very many has worked it out. So you take the photosynthetic reaction center and carefully plug out the chromophores that are present in it, and then replace it with whichever chromophores that we are interested in to put in back. And this has not been synthesized so far. It's an extremely challenging task, but the nearest that has been done is from Moore and co-workers from Arizona State University, where you take the components of the photosynthetic reaction center and just connect them one after the other in a linear manner. Whatever that is being attempted, it has not come closer to what is done in the photosynthetic reaction center. The only thing that we need to repeatedly realize is that, the photosynthetic reaction center, though well understood, it is extremely complex. And to have that complexity generated through synthesis is going to be extremely challenging. And definitely the protein scaffold, whatever that it offers in order to stabilize the charge separated states, perhaps cannot be done with anything else that are being decided in the area of multi-chromophoric assembly. But then, that is something to be left out in order to synthesize and try and see where do we go. But that's a very valid point.
Thank you. Thank you.
Okay. Are there any further questions? I'll just want to ask one. Mahesh, have you ever done in the crystalline lattice a resonance Raman experiment
No,
on these Greek key null aggregate motifs?
no. I mean, I can only request you to do that. I mean, I'm completely incapable of doing such measurements, but that would be really interesting.
Because it would actually show up as what modes are coupled to that exciton that has
I
been
fully
trapped.
agree with you. Yeah,
So,
I mean, those details are certainly a welcome thing, and we should.
yeah, so also it will talk about the delocalization with respect to the vibrations. What
Exactly.
delocalized vibrations it is coupled to.
Yeah,
That will be an interesting thing to observe because you have a very nicely homogeneous crystal
exactly.
where only one population is there of the cross, the Greek
Yeah.
cross. So maybe we can look at it. I mean, that would be very interesting.
And
see.
also this, electronic coupling being completely silenced, whatever that,
Right.
we can study, it will be quite
Exactly.
exciting.
Okay.
I fully understand.
Okay, Mahesh. Okay. Are there any other questions? Okay. Well, if there are none, let me actually thank Professor Mahesh Hariharan for a wonderful talk. Mahesh, this will be in our TIFR YouTube channel for a long time, and I request all of you to come back and view this again. And I would like to thank all the audience who joined in. This was the last talk of this year, and some of my colleagues from chemical sciences are there, of course, on physics as well. So if you guys, Mahesh, if you can unshare your slides, you can probably
Yes.
see them. If you can come up on the video, that would be great. It'll just be good way to sort of thank Mahesh. And also, Mahesh, if you were
Thank you.
here, you would have gone for tea and snacks.
Sure.
Unfortunately, you're not here, but for the first time after a long time, I've kept some tea and snacks outside. So for all the TIFR community members who want to come and join and eat some snacks outside the West Canteen, please do come out. I have some bit of snacks out there. So unfortunately, our speaker can't join, but at least due to the demands of the community across, I put something out there. So,
Thank you, Yudhishthira.
so yeah. So Mahesh, thanks. And next time I will have you over and for lunch and everything. Okay, dinner.
Sure. You should. You
That's
should.
around me. Okay.
Mahesh, can I just ask a quick question?
Yes, Deva, how are you?
Good, good. So it's regarding this, the null exciton, and have you looked at the photoconductivity of this material?
Not yet, not yet. And it's also not something which is of my skill sets. And certainly Upendra has agreed to look at it. So we are sharing.
And in terms of the temperature dependence of the, the lifetimes, as it were, maybe you showed a slide, but I missed it, or
Temperature dependence we have not done.
you've not done at all.
No, no. We are still.
What is your
trying-
expectation in terms of slightly elevated? I'm not talking about remarkably, 10, 20, 30 degrees above room temperature.
I don't know. I mean, there are two parameters that could be really exciting to look at. One is pressure, and see whether, you can really bring the chromophores little out of 90 degree and therefore start showing up the electronic coupling. And another one is temperature dependence, but then we have to make sure that the measurements done are accurate enough to make such claims, because we are not really doing it on one crystal, but instead it is a random distribution of several microcrystals. So these orientation effects have to be excluded, and temperature can always, just distribute the crystals. So we haven't done that yet, but thanks for the suggestion. We have to try that, both pressure and temperature. Yes.
Okay.
There's another little small thing that is going on currently under the battery and supercap community, and this is about just storage of that excited state. So
Okay.
in your case, you don't have any redox active sites, but you could have capacitive. I'm
Yeah.
not sure, in terms of a type of a capacitive charged state that you can sort of hold on to.
Yeah.
Prolonged periods.
Yeah.
So
Yeah.
it comes under the word of persistent photoconductivity, if you want to.
Yeah, this is something I have heard our colleague Shaijumon talking about. So currently, I don't have much idea about the battery applications, but certainly these are structures that can be modified to look into.
Okay.
Very
But any little thing you do, these kind of structures are so sensitive, so they get just shattered. That's it.
I suppose so. I mean, I come from a different, so unless we try and play with them, and
Yeah, even an alkyl chain or a methyl group you move, then you don't get such kind of orientation.
Okay.
Yeah.
Okay.
Very nice talk.
Thank you,
Thank you.
Deepa. Thank you for the
Yeah.
question. Ravi?
JD, so I just wanted to, because I actually had a student coming in towards the end, so I missed the question answer session actually, unfortunately. But if I can just talk to Mahesh for a bit, is that okay? Are you getting late for the tea or something?
Yes. So let me conclude the session. I'll keep the Zoom on.
Yeah. Then I will
You can have a
leave.
brief chat. No problem.
I will leave.
Mahesh, we are going for tea in your talk celebration. Unfortunately, you are not there. We are pretty selfish. Sorry about that. But the next time you are here, I definitely promise you more than tea.
Yes.
Okay.
Definitely.
Okay. Thanks.
It's okay.
I want to thank. Okay, before Ravi launches into, I want to thank the auditorium staff here for putting this through. For people to come in and view the talk in the lecture theater, even in the absence of the speaker. So thank you, Mistriji, and thank you, Najapa. And I also would like to say that we will return next year, right on 5th of January, with Professor Yamuna Krishnan joining us from the University of Chicago. Okay, so
Chicago or Harvard?
University of Chicago still. Okay.
Okay.
Okay. Thank you guys, and I'll stop the